Hi, welcome. This is the 87th Cold Spring Harbor Symposium on Quantitative Biology, and the subject this year is stem cells. And I'm very pleased to have Jürgen Knoblitch with me, who spoke yesterday in the opening session.
That's right.
So you're using iPS cells to model or make organoids for using in modeling human development, neurodevelopmental disorders.
That's correct. Yes.
What particular disorders are you interested in and looking at?
So our philosophy is that by studying human brain disorders, we can learn something about the human-specific features of brain development, which is really what we are interested in. And then we can take that knowledge and develop some understanding of the disease, which will help the patients. So the technology that we are using actually goes way back. Some of it was developed by Yoshiki Sasai, but we were the first ones to actually realize that one can combine this with another technology, which is iPS cell technology. And when I say we, I mean Madeleine Lancaster, who was a postdoc in my lab, and what we decided to do was to model microcephaly in organoids. Microcephaly is a very severe neurodevelopmental disorder where the patients suffer from loss of brain volume. And of course, that leads to very, very strong mental defects. And by using iPS cells from patients who suffer from the disease, we could generate organoids that were much smaller. And we noticed that, okay, we can model the disease. And then we could go back and we could investigate the reason for why these brains are so smaller. And to our surprise, we found that it is not just because the cells proliferate less, but because they switch from a symmetric division mode to an asymmetric division mode prematurely. So in our brain, just like in the mouse brain, cells, the progenitor cells, first expand by symmetric division. So 2 make 4, 8, 16. And that goes on for much longer in the human brain than it does in the mouse brain. And that's how you generate first a large number of progenitor cells, and only then you switch to an asymmetric division mode and start generating neurons. And this requires that the orientation of the division plane is correctly specified. And we found that in those patients there was a defect in the centrosome integrity, so they cannot orient their mitotic spindle. They cannot divide in the right orientation, and as a consequence, they cannot sustain these very long symmetric divisions. And that's why we were able to learn something about the disorder.
And that was previously unknown, certainly unexpected, unknown.
You could, so for this particular disorder, it was unknown. If you go into the literature, the role of the gene that was affected in spindle orientation was known. So I wouldn't say that this was the, I wouldn't say that the learning something about the disorder was the big breakthrough. I think the big breakthrough came because we were really the very first ones that used three-dimensional culture grown from human iPS cells to model a human disease. And this is why the field afterwards really exploded. And I guess this is why science voted this as one of the top 10 discoveries that year.
So you've now demonstrated that one could use such organoids to explore a human neurodevelopmental disorder. And after that first success, what have you gone on to look at?
So first of all, I would like to give credit to also other people. There were many other labs. I think probably one of the other very big successes of the organoid field was work from Guo Liming and Hongjung Song, who used it to demonstrate that the Zika virus can cause microcephaly. You probably know that the last time the World Health Organization called a global state of emergency was in when the Zika virus broke out in Brazil, and at the same time there were many children who were diagnosed with microcephaly. But it was really the organoid experiments that showed that the Zika virus can cause microcephaly. And since then, many different disorders. So there have been cases of epilepsy, cases of autism. We have modeled tumorigenesis in organoids. We have modeled different kinds of structural neurodevelopmental disorders. Most of them, I have to say, were confirming what people have already known. And now I think the field is switching. It has gained enough confidence to do experiments that are challenging the existing views, which I think is really what we should do.
Can you give an example?
So we have recently studied a disorder that's called tuberous sclerosis. It's a severe form of epilepsy, and it results from mutations in either TSC1 and TSC2. So these two proteins inhibit the mTOR pathway, which is a major regulator of proliferation and cell growth. So if you have mutations in those, the cells proliferate more and they grow more. Now the patients have only one copy of TSC2 mutant. When you make a mouse that has one copy of TSC mutant, absolutely nothing happens. The mouse is completely, has no defect. And so people thought, well, you know, because what we learn from mice must be transferable to humans, what must be happening in the human patients is that they must have a spontaneous mutation that inactivates the second allele. Because if you do that in the mouse, if you mutate the second allele, now you get all kinds of defects that are very similar to what you actually see in the patients, although you could never really recapitulate the disorder. And we did not really want to believe this. When I say we, I actually talk about a medical student in my lab, Oliver Eichmüller, who was courageous enough, initially against my advice, to actually just grow organoids for very long. And we had organoids from TSC patients, and what we noticed was that they form bumps on the surface. We then looked at these bumps, and these bumps were benign tumors, and that's exactly what the patients suffer from. The patients also have so-called tubers. Tubers are inclusions in the brain that you can see in MRI, and they consist of very large cells, so-called giant neurons. And they are thought to be the epileptic cores that cause epilepsy in those patients. And when we change culture conditions, we can also model that. And to our surprise, we noticed that both of these alterations do not require a loss of the second allele. So in humans, there is a certain cell population, which we found by single-cell transcriptomics, that is very sensitive and that reacts already to the loss of one allele of TSC2. And that, of course, challenged the entire field. I think initially it was not easy to publish this, but I think now it is actually accepted in the field that the mechanism of tuberous sclerosis development in humans is different from what exactly we were predicting from mouse. And that led us to discover this cell type, which we call the clip cells. And they do all kinds of interesting things. We think they are responsible for certain things that are happening only in the human brain. For example, after birth, in a human child, there's a massive migration of interneurons into the cortex. This doesn't happen in the mouse, and we think these interneurons come from these clip cells. And so this now allows us to study these events in organoids.
Which I suppose makes me then think of the, what was it that Jacques said, what's true of an elephant is true of man. But clearly that's not the case in the ones we've just been discussing about mice and human beings.
So,
so is that being able to use human organoids of whatever type is able to circumvent that? You're now able to do experiments in humans, so to speak.
Yeah, so you will be surprised to hear this, but I'm actually one of the big skeptics. So there is a lot of stuff out there about brain organoids that exaggerate it. People, they call them mini brains, and they say that they will replace computers. And I feel, I'm really sorry. I think this is just nonsense, right? But they're a reductionist model. I mean, they are lacking blood vessels, they are lacking microglia, they are, you know, not like a real brain. But they have human genetics. So you can take cells from a patient and the organoids are genetically identical to that specific patient. And you can ask, what is actually happening when a human has a particular genotype, a particular genetic aberration? And then you can learn something. You can learn something which then, you know, you might need to test in the mouse model. So I think organoids will help us to understand the human brain better. I'm a bit less optimistic whether they will completely replace animal experiments. They might allow us to do them in a more targeted way, but I think their real advantage is that they're a new model system. Just at some point, zebrafish was started as a new model system, and now it's here, and it's here for good, and it's a fantastic system. So now organoids are an additional one, and they're complementing rather than replacing the other models.
Well, maybe we should stop on that optimistic note. Thank you very much, Jürgen.
Thank you.